Okay, so the next topic for this unit, biotechnology, it is gel electrophoresis. Okay, gel electrophoresis. So in this, it is the process. Okay, this gel electrophoresis, it is the process of separation of DNA fragments. Okay, it is the process where DNA fragments are separated based on filtration in the gel like substance. Okay, based on gel filtration as well as electrophoric mobility of the DNA molecule. Okay, electrophoric mobility means when electric current is passed to the DNA molecule, it moves towards positive charge. Okay, why? Because DNA is negatively charged molecule, so when electric current is passed through it, DNA moves to the positive charged, okay, positively charged electrode. Because you know that like charge repel and opposite charge, they attract each other. Okay, so the steps involved in this gel electrophoresis, how it is done. Okay, how the fragments of DNA are separated on the basis of gel filtration and electrophoric mobility of DNA. The steps involved are number one is isolation of DNA. First of all, DNA needs to be isolated. Okay, we need to extract the DNA that we wanted. Okay, then after that, number two is DNA. Since you know that DNA is very long, okay, very long chain. Okay, DNA is very long chain, so we need to make the fragments of DNA. We need to make different fragments of DNA. So for that, restriction digestion means separation of this DNA. Okay, making fragments of DNA. This DNA, long chain of DNA, it needs to be broken down into smaller fragments. For the same reason, the enzyme which is called restriction endonuclease, you already know that enzyme is exposed to this DNA so that it can be made into different fragments. Okay, now after the fragments are made, now the fragments of DNA, they are different. Okay, different sized fragments of DNA. Some are small, some are long fragments. So this is loaded in a loading well. Okay, this is loaded in a loading well. This DNA fragment is loaded in a loading well. Now what happens after it is loaded in a loading well? Now this loading well has cathode and anode. Okay, this loading well has cathode and anode. You know that cathode is electrode, it's a rod. Okay, electrode which is negatively charged. Cathode is negatively charged, whereas anode is positively charged. Okay, don't get confused. Cation is positive charge, ion, it's an ion. Okay, positively charged ion, but cathode is a negatively charged rod, that is electrode. Similarly, anion, it is negative charge ion, but anode is positively charged rod, electrode. Okay, so there are two electrode, cathode and anode, and this restriction DNA digested, restriction digested fragments of DNA are loaded in a well towards the cathode, towards negatively charged cathode. Okay, rod. Now what happens is, when DNA is loaded towards the cathode, you know that DNA is a molecule which is negatively charged. Okay, DNA is negatively charged molecule. So when it is loaded towards the cathode, it cannot stay there for long, because cathode itself is negative rod. Okay, and this molecule is negatively charged, so it moved towards the anode. Okay, positive charge. This negatively charged molecule of DNA, they move towards the positively charged anode. Now here the negatively charged DNA molecule move towards anode under the electric field. Now, when it moves, when this well is supplied with the electric current, it cannot move on its own. Okay, the molecule cannot move on its own, but in this loading well, when the external electric field is supplied, electric current is supplied, at that time this negatively charged DNA move towards the anode. Okay. Now, one thing remember, the smaller fragments, because this is different sized fragments, it is not same. So, in this the smaller sized molecule move faster, okay, travel longer distance through the gel. Gel is kept, okay, one gel like substance is kept in this well where the DNA molecule is loaded, okay, gel like substance is kept. Now, a smaller molecule move faster, travel longer distance through the gel than the larger fragments, okay. So, at first smaller molecule will be there and then large molecule like this. It takes more time. The large molecule, large fragments will take more time, whereas a smaller molecule, smaller fragments will take shorter time because it moves faster. Alright. Now, these are separated. So, the separated DNA fragment is transformed to nitrocellulose paper. Now, when it is separated here, then electric current is switched off. Okay. Now, when it is separated, the fragments is separated, these fragments, okay, the same fragments, okay, when they are separated, it is transferred to nitrocellulose paper. Okay. Paper is kept on this gel so that the fragments, this nitrocellulose paper, so that the fragments of DNA is transferred to that paper, nitrocellulose paper, okay. And then the same nitrocellulose paper is blotted. It is blotted, okay, with ethidium bromide. It is blotted with ethidium bromide. Why? In order to stain that nitrocellulose paper, because DNA is not colorful. Okay. So, in order to see the different sized fragments of DNA in nitrocellulose paper, the paper needs to be blotted with ethidium bromide, which will stain the DNA molecule. Okay. Now, after that, the next step is expose this nitrocellulose paper to UV light. After it is blotted with ethidium bromide, the nitrocellulose paper is exposed to UV light. Now, when it is exposed to UV light, the stained DNA, now the DNA is colorful, okay, because it is stained with ethidium bromide. The colored DNA shows bright color bands. It shows bright color bands like this. Okay, it shows bright color bands. These DNA fragments will show bright color bands in the nitrocellulose paper when it is exposed to UV light. Now, cut the separated bands of DNA and extract pure fragments of DNA to use it for PCR. Now, the same DNA, it can be removed off from this nitrocellulose paper and it can be used for different biotechnological processes. Okay. So, this is all about gel electrophoresis. Here, in the loading well, gel is also kept where the DNA molecules are loaded at first, then electric field is supplied into this well. When electric field is supplied, the negatively charged DNA move towards anode, the smaller fragments move faster, okay, then it is transferred to the nitrocellulose paper. After that, it is blotted with ethidium bromide and then UV light is exposed to that nitrocellulose paper so that our DNA will be visible. It shows bright color. Okay. So, this is all about gel electrophoresis. Okay. So, now gel electrophoresis is understood. Now, the next we have is DNA finger printing. DNA fingerprinting. Now, It is a technique of determining nucleotide sequence of certain areas of DNA which are unique to each individual. Okay. So, it is a technique, this DNA fingerprinting, it is a technique of determining nucleotide sequence. Nucleotide sequence is present in DNA, you already know that. So, in order to determine the type of nucleotide sequence in DNA, okay, which are unique to different individual. Now, what are the steps involved in DNA fragments? At first, extract the DNA from the cell. We need to extract the DNA, that means we need to isolate or extract the DNA from a cell. From a cell, any cell, it can be blood cell, saliva, whatever, okay, any cell. Now, after that, again restriction digestion in order to make different fragments, okay, restriction digestion to generate restriction fragments. Now, when this long DNA, it is digested, it forms smaller segment. Now, every individual have the fragments, smaller fragments, every individual have V and TR, that is variable number of tandem repeat. That means in nucleotide sequence, suppose this is adenine, thymine, T, G, A, like that. So, here you can see adenine is repeated, okay, G is repeated, T is repeated. So, this kind of repetitive sequence, okay, suppose G, G, A, C, C, G, G, again like that, again A, it is like that. Then this nitrogenous base, guanine, 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 it is repeated several times, okay. So, this type of repeated sequence, it is called VNTR, VNTR, which means variable number of tandem repeat, variable number of tandem repeat, okay. So, restriction digestion to generate restriction fragments. So, restriction fragments shows it contain variable number of tandem repeat, okay. Now, again number 3 is separate the fragments. We need to separate different sized fragment, because after restriction digestion the fragments can be of different sized, okay. So, we need to separate these fragments into fragments at first, and how we can separate that? Through this process, that is gel electrophoresis. So, third process is gel electrophoresis in order to separate the fragments, okay, in order to separate the fragments. Now, transfer the separated fragments from gel to nitrocellulose paper by blotting the paper over the gel. So, in this gel electrophoresis, I have told you that from this well, okay, from this loading well, from this gel, okay, it is transferred to the separated fragments of DNA is transferred to nitrocellulose paper, and the same process, okay, the same process is called Southern blotting, okay, this process is called Southern blotting, okay. So, now after this, after it is transferred to nitrocellulose paper, what happens is that we need to do that synthesis special DNA probes containing repeated sequences complementary to those on VNTR. Probes are radiolabeled with radioisotopes, very important. So, after electrophoresis, we need to synthesize, that means artificially we need to synthesize special DNA probes. Now, DNA probes means in a lab, okay, DNA probes means if the fragment is having, suppose if I unwind this, if the fragment is having A, T, 5' to 3', 5' to 3', A, T, G, A, like that, then the probe, DNA probe which is synthesized in the lab should have T, A, C, T, like that, okay, smaller fragments. So, that is DNA probes. It should be complementary to the DNA, and if it is opposite to A will be T, A, C, T, then the probe will have A, T, G and A. So, that is DNA probes, okay, just complementary to that of that fragments, DNA fragments. So, synthesize special DNA probes containing repeated sequences complementary to those on VNTR, okay, complementary to those on VNTRs. And these DNA probes which is synthesized in the lab, they are radio labeled, okay, with radioisotopes. Now, why they are radio labeled? In order to hybridize the DNA probe with that of this VNTR fragments, these DNA fragments. In order to hybridize the DNA probe that is synthesized in the lab with that of the DNA fragments already there, okay, the DNA probe should be radio labeled with radioisotopes, okay. All right. Now, radio labeled probe is allowed to hybridize with DNA fixed in nitrocellulose member through VNTR locus. So, now they are allowed, this DNA probe, they are allowed to hybridize with that of DNA already present in that nitrocellulose paper, with that of DNA already present in that nitrocellulose paper. Now, after that, expose X-ray, okay, films to the nitrocellulose membrane. So, this nitrocellulose paper now which is hybridized with the DNA probe, okay, these DNA fragments now is hybridized with the DNA probe. After that, it is exposed to X radiation, okay. When it is exposed to X radiation, dark band appears when X-ray film develop, indicating the position of DNA fragments. So, when X-ray film it is produced, this DNA fragment which is hybridized with the DNA probe, dark band will appear in that position. Dark band appear like that in this X radiation film, in this X-ray film, okay, dark band appears like this, indicating the position of DNA, okay. And if same is compared with that of other members, suspected members, then some may have like this, one here, one there, two there and one here. And if the other person, suppose this is person number 1, the other person, okay, suspected person from the crime site, suppose if it has like this, then DNA fingerprinting is matching to that of the person B. So, this will be our criminal. So, this way DNA fingerprinting is done in order to find out the criminals from the crime site, okay. I will give you some example. If this position of DNA, okay, DNA fragments, it appears when if there is any crime site, okay, and from that crime site if blood is taken out from that crime site, if anybody have left the blood drop in that crime site, and that from that blood cell this DNA fingerprinting is done, which shows the band like this, okay, position of band like this. And if the police is suspecting 2 person, okay, 1 person, 2 person, then from that person also any cell, saliva cell, blood cell, any cell, cell from these 2 person are taken out, and then again DNA fingerprinting is done with that cells, with that DNA, okay. Then if first person's DNA fragment is showing position like this and the second person is showing position like this DNA, then our suspect 2 will be the criminal, because it is matching with that of the sample that we collected from the crime site. So, this way how DNA fingerprinting is applied. Okay. All right. So, this process now I forgot to mention one thing, the process of exposing this X-ray film to the nitrocellulose paper in order to find out this position of DNA fragments. Okay. This process, it is called autoradiography. This process is called autoradiography. Okay. Where X-ray films are exposed to the nitrocellulose paper, which will give us the position of DNA fragments, position of separated DNA fragments. Now, what are the application of DNA fingerprinting? Now, how it is used? I have already told you. DNA fingerprinting is used in criminal identification like this, solving paternity and maternity disputes. Okay, that means whether the child belongs to that father or mother or not. Okay, that is paternity and maternity disputes. It also help in determining evolutionary history between the two organisms. Okay, if one species is very relative to the other species, we cannot simply say that yes, it is related and maybe their ancestral species are same. We need to trace this DNA also, DNA history. If the DNA is more or less matching, not 100%, but at least 40 to 50% they are matching, then we can say that yes, these two species are quite related. Okay, so that is evolutionary history. So, this is all about applications, okay, where DNA fingerprinting is applied. Please don't get confused. Fingerprinting does not mean that printing with the fingers and all. It is not like that. The name is DNA fingerprinting, but it is all about DNA, not fingers. Okay, it is all about DNA. So, this is all about DNA fingerprinting and gel electrophoresis. I hope it is clear now. Thank you.